Hey guys, welcome back. This is Lei. In this video, I'm going to show you how to adjust for batch effects in RNA-seq analysis. So what are batch effects? Batch effects are unwanted technical variation in gene expression. And what is variation? Variation is a change of gene expression across groups. It's a difference we want to detect during differential expression analysis. However, batch effects can confound the results of differential expression analysis. For example, in this study, colors represent batches, shapes represent the groups. Samples in each group are sequenced independently, so we have 3 batches and 3 groups. In the PCA plot, the samples are clustered by groups as well as by batches. So it's impossible to tell if the differences between the groups are caused by biological conditions or just by batches. It's completely confounded. This is the worst study design. Of course, the best study design is to process all the samples at the same time by the same person on the same day and sequence them on a same plate. In this way, there will be no batch effects at all. However, in reality, the most commonly used study design is the balanced study design. In this design, samples are mixed and then are sequenced in different batches. In the PCA plot, samples are most likely clustered by batches, and ideally we'll be able to remove the batch effects and only preserve the differences between the biological conditions. And this is the topic for today's tutorial. The batch effects adjustment can be done in R environment. The first step is to set up your R environment. Open the R and RStudio, install all the required packages, and load them. The example dataset can be downloaded from the ComBat-seq GitHub and can also be downloaded from my website, ngs101.com. The dataset is in the format of R object. You can read R object using the readRDS function. And once read in, we can extract the gene expression matrix from the object. You can see the object as folders and subfolders. So we access the subfolders using the @ sign, and there's a subfolder called data, and in the data subfolder there's a data matrix called counts, and this is a gene expression matrix like this one. For the sample information table, we also retrieve it from the R object using the @ sign. Here is a meta table for sample information. And then we changed the batch column into factor using the as.factor function. And next, we just sample the groups to create a simulated treatment variable. And then we do some QC steps to remove the low expressed genes in our gene expression matrix. Now let's visualize the batch effects using the PCA plot. And I've showed you how to make PCA plots using the gene expression matrix before, so I'm not going to repeat it again, but we just transpose the gene expression matrix and turn it into a data frame and feed the data frame to the PRComp function and use the autoplot to plot the PCA plot as I showed you before. As you can see, the samples are clustered clearly by the 3 batches, and they are widely separated. I'll show you 4 different ways to remove the batch effects. Let's start from the ComBat-seq. From the SVA package. So we're going to use the ComBat-seq function. This function accepts 3 inputs. 1, expression matrix, the batch information from the sample information table, and the treatment information. So the rationale is to remove the batch effects and preserve the treatment information. So the function will return another gene expression matrix in the same format, but with the different values. And then we'll make another PCA plot to visualize the results. As you can see, the samples from different batches are mixed together much better than the one before. So that means we have removed a lot of batch effects from our data. The next method is from limma. That's why I like limma, because limma is so versatile. It can do a lot of things. So as usual, in limma, we have to create a DG list object by combining the gene expression matrix and the meta table. Then we normalize the gene expression data and call another function called removeBatchEffect. This function accepts the gene expression matrix and the batch information and returns another gene expression matrix with different values. With the correct gene expression values, we can make another PCA plot in the same way. And as you can see, the results are much better than the one we used before, the ComBat-seq. In this PCA plot, all the samples are very well mixed, much better than before. And the next one is the mixed linear models. And this method is quite different. And we're going to use a linear model to regress out the batch effects, because there is no function for that. So we're going to make our own function. The function will accept the gene expression table and the meta table. It's a sample information table. We put the gene expression in a mixed linear model in this format. That means we're gonna model the treatment and the batches, and we got also feed the metadata into the limma, the linear model. And next we take the residuals and add the average expression of the entire dataset to make a new expression value. Because the function only works for one gene, so we have to loop through the entire gene expression matrix. So we call the function using apply. Apply is a loop, vectorized loop, to loop the function through the entire dataset for all 20,000 genes. And then we convert back to the same format as the original gene expression matrix. And then we make the PCA plot as we did before. As you can see, the results are pretty good. All the samples are mixed well, and you can't even tell the shapes of the different batches. So the rationale is like this. We model the treatment and as well as the batch effects, and we only take the residual. What is residual? It's the predict value on the regression line minus the original value. The residual will have no correlation with the original gene expression value. So we'll take the residual, add the average expression, which is the interceptor in this regression model. We use this new value as our new corrected gene expression value. This linear model regress out the batch effects as well as our treatment information. So if possible, I don't recommend using the mixed linear model, except you know that the batch effects in your dataset is so strong and is so dominant, so we can use this mixed linear model. So the last method for correcting batch effects is correct the batch effects in the differential expression analysis. Here I introduce the way to regress out the batch effects in 3 D packages. In DSeq2, we just call the same function. The only difference is we put the batch effects in the design matrix, and then we call the function and do the regular differential expression analysis. And for limma and HR, it's the same. Everything is the same, except in the design matrix, we put the batch effect in the design matrix. And the model will take care of the batch effects and adjust for the effects during the differential expression analysis. So for all these 4 methods, this is actually the best way to adjust for batch effects. So if you want to use gene expression for other purpose, you can use either ComBat-seq or limma, but be cautious to use mixed linear model for batch effects adjustment. And that's all 4 ways to adjust for batch effects. Use this one if you are only doing differential expression analysis. And for more details, go to my website, ngs101.com. And if you liked it, subscribe, and I will see you in the next video.